Dear friends, so today we will be discussing about homology dependent DNA repair, which is abbreviated as HDR. Such kind of repair is dependent upon the homologous chromosomes to generate the lost part of DNA. This kind of repair takes the help of homologous chromosomes, or it can take the help of the sister chromatid also. That means we just need a strand of DNA that has got the same sequence of nucleotides as are present in the defected strand. So let's see how it occurs. Suppose this is our DNA, intact DNA. It has got two strands, one is 3' to 5' direction and the other one is anti-parallel from 5' to 3' direction. Now due to some ionizing radiation or X-rays or chemotherapeutic agents, free radicals also, due to any of this kind of insult, sometimes we get a break in the DNA molecule such that both the strands of the DNA molecules are cut. So this is the break. Now this was intact DNA molecule, it has been cut at both the strands. Now what happens? This mRNA complex comes here. This is the main complex that plays a crucial part in HDR. mRNA complex is actually made up of three proteins. One is M or MRT2 or MRE2, second is R or RAD50 and the third is N or NBS. This mRNA complex comes and bind to the 5' end of the broken strands. In this strand, this was 5' end and mRNA complex has come and got bound here. For this strand, this was the 5' end and mRNA complex has come here. So what is the main function of mRNA complex? This complex comes and gets bound to the 5' end and then it keeps on reducing the length of this strand. So it keeps on cutting the nucleotides here, so that ultimately this end becomes shortened and we get a 3' overhangs here. Let's see the process till now. This was our intact DNA molecule. Due to some kind of insult, both the strands of the DNA molecule has been cut here. Now mRNA complex recognizes the 5' ends and comes and gets bound here, and then it keeps on removing the nucleotides from 5' end, so that ultimately this 5' end gets shortened, this strand gets shortened. In the other strand we have got the 3' overhangs. See, this length has been shortened, so ultimately this strand becomes longer than this one and we have got the 3' overhangs here. So now we have got this chromosome, this one 3 to 5' direction and the other strand is from 5' to 3' direction, where both its strands has been broken due to some kind of insult. So what happens now? So now first we have to ensure that these 3' overhangs does not coil back to the other strand. So for that, replication protein A comes that protects this 3' overhang against the action of nucleases, as well as it prevents the coiling back of this 3' overhangs. Similarly in the other side also, RPA proteins, see these are RPA proteins, that is the replication protein A. So this 3' overhang also is protected against nucleases with the help of RPA protein, as well as its coiling back is also prevented by the RPA proteins. After this, one more protein, that is BRCA2 or breast carcinoma protein 2 comes. This protein binds at the junction of double-stranded DNA with single-stranded DNA. See, till this portion, this DNA molecule is in the form of a double strand and after this portion it's in the form of a single strand where already RPA proteins have been attached. So BRCA protein comes and gets attached at the junction of double stranded DNA molecule with the single stranded portion. Then BRCA protein helps the RAD51 proteins to bind at the same spot. This RAD51 protein can only bind to the single stranded DNA, and this protein helps in the search for the homologous DNA. So when these two proteins are here, BRCA2 as well as RAD51, RPA protein gets removed. Now there is no danger of the chain to coil back upon itself. Now this RAD51 protein helps the DNA molecule to search for the homologous chromosomes. So let's see here. Now RPA proteins have been removed. RAD51 is there. RAD51 helps this DNA molecule to search for the homologous chromosome. See, this is the homologous chromosome. This protein has come here and this is the complementary strand. See, this strand is going from 3' to 5' direction, and our template strand is from 5' to 3' direction. So this strand is complementary to the nucleotide sequence to this strand. See, this strand and this strand, they are complementary to each other. Now new nucleotides are attached here according to the sequence present in the homologous DNA molecule. These are the new nucleotides which have got attached here with the help of DNA polymerase enzyme. So this new chain formation actually results in the formation of a displacement loop. This displacement loop is formed in the homologous chromosome due to the unwinding of the homologous chromosome and incorporation of a new strand in the homologous chromosome. This displacement loop keeps on moving as newer and newer nucleotides are getting attached here. Ultimately, all the lost nucleotides gets attached here. Then with the help of enzyme DNA ligase, this strand gets attached to its parent remaining strand. See, this was our template strand. These are the new nucleotides which have been got attached, and with the help of DNA ligase we have got the complete new strand here. So the rest process is easy. This is our 3' overhang in the other strand. Now same sequence occurs. These RPA proteins gets removed, and as in the process of DNA replication, with the help of enzyme DNA polymerase and DNA ligase, it's very easy to get the new nucleotides attached here according to the sequence present here. So when all the nucleotides gets attached here, then with the help of DNA ligase, these two ends are ligated. So ultimately we get the complete new DNA molecule even when both its strands have been broken with the help of the sequences present in the complementary strand. So in a nutshell we can say, in this process, with the help of RPA proteins, BRCA2 protein, RAD51 protein, and using the homologous chromosome as the template strand, our body can make the complete new DNA molecule. So this is how DNA repair by homologous recombination occurs. It actually occurs during the synthesis, during the cell division phase.